The overall goal of this assay is to determine any changes in fat storage levels that may be caused by a change in genetics, diet, or treatment.
This method can help answer key questions in the field of metabolism, such as genetic or neuronal contributions to obesity predisposition. The main advantage of this technique is that it's fast, economical, and very reproducible when compared to other methods currently used.
Individuals new to this method or new to working with wandering stage larvae may struggle with this protocol because the developmental timeline of different genotypes can be highly variable.
To begin this procedure, add a small dollop of yeast paste to the middle of a grape plate. Smooth down any rough edges with a spatula. Next, transfer the flies of interest into an egg collection chamber and place the grape plate with yeast paste on top facing inwards. Then, tape the grape plate to the egg collection chamber to keep it from falling out. Make sure to write important genotype and date information on the bottom of the grape plate. Subsequently, upend the egg collection chamber to allow the flies to lay eggs on the grape plate. After that, cover the egg collection chamber with an open box and place the covered egg collection chamber in an incubator at 25 degrees Celsius for 4 to 6 hours to allow the flies to lay eggs. When it is done, take the grape plate out of the egg collection chamber and transfer the flies back into their original bottle of food. Use a spatula to wipe off any excess yeast paste from the grape plate.